Okay, my friends, it's a new day at this university, and we are going to start out with chemistry, biochemistry, the type of chemistry that's in your body, the type of chemistry that makes you live and breathe. And this ligands, I will explain to you how important they are. And these are the things that attach to the molecules in your body, and they transfer all through your body and deliver and pick up other molecules. We're going to go over that in a second. And I want to make you comfortable with the fact that I understand this, I think, as well. And I actually, to be honest with you, I know it sounds like I'm bragging, but I think I understand it better than anybody else right now. Because the Bohr model of atomic theory is totally proven wrong. The nucleus is nothing like they said. And therefore, they can't even account for gravity. Because with the current model, it doesn't work with anything. So, but they can go up to wormholes and make all these theories because they can just override the fact they don't understand what the atom is constructed of. So we're going to go into that deeply. I mean deeply. And it is simple. Once you understand it, it's just a kid can understand. If you're 10 or 12, you're all set. You have no problem whatsoever understanding this. Very, very, very simple. I do want to show you some things that are going to look extremely complex. And they are. But you won't have to know any of this stuff. I just want you to understand that I do know it, and I'm having a fabulous time. I'm really enjoying this because this was I loved this stuff, and I still do. And I hadn't had a chance to do this, business and so forth. Now this is all about oxidation reduction and how molecules and atoms interact to make different substances. And I did this a lot. I mean, and this is about thermochemistry. This is where things change. Now, if you ever worked with concrete, and you put the stuff together in the water and all this, all of a sudden it gets hot. Well, what's that all about? Why does it get hot? And why does it all of a sudden get hard? Well, what's happening is it's forming bonds and binding. And when you bind something and bond something, you a lot of times liberate extra electrons. It's all it is. Because that's all there is electrons. Now I'm going to explain that to you because the electrons build together to make the protons and so forth. But as these molecules in the water and all this, boom, they come together, they got to spit out a bunch of electrons. Where do they go? Well, they're like balls of energy that say, well, who wants me? And they said, nobody wants you, get out of here. And so they start pushing, and they're heat, and they start to flow into the colder areas that will accept them, because they'll say, well, I'll take you more than that guy, so come on out here. And that's what happens. They drift off. That's what heat is. And that's what cold is. The cold is where it wants electrons. The heat is where get the hell out of here. So this is thermochemistry. I love this stuff. I mean, I did a ton of this stuff. And anyway, so if you feel comfortable, I do understand what I'm talking about here. Now, and then I also did the actual, experiments, or heat of solution, heat of reaction, calorimeters, and neutralization, all this stuff. And then it all ends up giving off heat, and heat can be transferred into electrons. And then once you get into electrons, you get in, are those electrons being delivered? Then you got into serial and parallel and all that. And I understand all that too, and you're going to understand it. It's very, very simple. It's absolutely simple. It's just like water going through a hose. You won't believe how simple this stuff is. Anyway, we're going to do this, and you're going to learn from scratch. We're going to start at the beginning, and you're going to understand this. You understand this is CNOS, sulfur, oxygen, carbon, nitrogen. This is life over here. Really, that's life. And these are the things that make the life transfer in and transfer out. You got your salty things and your acid things, and they push things back and pull things forward. They add electrons, subtract electrons. That is the flow of life. All right? continuously, as food enters your digestive system, it is being transmuted. And it's being transmuted by bacterial enzymes. They are little chemistry sets that go in and crack molecules and say, break that one and add it to this one over here and take two of these over here. And that's what gives you the energy to do things and to build your body, to support your structure and all that stuff. So we're going to get into this. But ligands, what I want you to see up here, ligands are your friend. These are the things that make you work. And it's let's just read it. A ligand looks like this. It's a whole squiggly bunch of little molecules, right? And this is the heme, which is your hemoglobin. And that's the thing that carries a ton of oxygens. I'm going to show you that because iron is in here. Iron has a natural affinity to go out and grab a hold of oxygens. Oxygens have binding sites. Let's look at this. The binding sites carry other things around. That's my point. And the iron is in the blood cells. The binding sites are on the oxygens. The oxygens carry the other molecules, which are your transition metal complexes. They contain all, basically, of the things that you need for life: nutrition, oxygen, all of the different molecules, and hydrogen, every single different gas and everything. And they carry it through your bloodstream, and when they get to a place where it has an electron that is, or whatever, it has a chemistry there that favors it, it'll jump over. That's all. And it'll go to the place where it's needed to construct something or to add energy or to do whatever it's supposed to do. And there's a lot of neurotransmitter chemicals. This is where it's key. This is the problem with autism and so forth. I've focused in on the neurotransmitter molecules. And these are also part of the breakdown of products that are in your body by bacteria. It's an enzymatic breakdown. It's a reductase. It takes these things and it breaks them up in little bits and pieces. Okay, you need this, here it is. Here's some of this. You need that, here's some of this. I mean, it really has got something. It's got to almost be that way. It's got to be there's some control in there that says, what do we need to be healthy? And it says, well, I need some sulfur. I'd like a little of this. I'd like a little of that. And something makes it happen. And it only happens by these enzymes. And it only happens by the enzymes being produced by the bacteria. And it only happens if the bacteria is alive to produce it in your body. And something has to be talking to that bacteria in your vagus nerve or something. That's a whole new ballpark. But until you can get to that ballpark, you got to get through this ballpark. And this one says, here's the way I move through your body. I move products through your body with these ligand attachments. All right, so I ran off at the mouth, but we got plenty of time as far as I'm concerned. Now, so what is in biochemistry and pharmacology, which because they use them in drugs too to attach to things. A ligand is a substance that forms a complex with a biomolecule. It's a living molecule or some molecule that does a biological thing to serve a biological purpose. Like oxygen for you, removing carbon dioxide and all that stuff. In protein ligand binding, the ligand is usually a molecule which produces a signal. Don't forget this. By binding to a site on a target protein. It creates a signal by binding to a site on a target protein, So the binding typically results in a charge of conformational isomerism, which is conformation of the target protein. So it conforms it. Changes. In DNA ligand binding studies, the ligand can be a small molecule ion which is an ion, or a protein, which binds to the DNA double helix. So you got your DNA strand comes down. This little ion comes over. It snags a spot on the double helix. Now, in my world, that is methylation. And I've gone over that, and I have a whole new story on methylation that I just realized about neurotoxin methylmercury. Methylation is your brain's programming. So, whoops. All right. The instance of binding occurs, well, anyway, let me go back to this. The binding typically results in a charge of conformational isomerism, which is the change in a target spot on a protein. And that is, I can really get deep into this, and I will, and I have. But what it does is it turns it on and off. There's a chunk of DNA that relates to something, a process, let's call it. All right, and I understand this too, because I did a lot of stuff. I'm not bragging. I just did a lot of stuff. I understand programming. What you do is you have a process section, and it's either on or off. And if you say, I want to turn this on, it goes out and does the job. In here, it's the sequence of DNA that says if there's a C and a G, all right, the CTAGs. CG is your methylation molecules. And if it's methylated, it's on, or if it's unmethylated, it's off. It's as simple as that. It's a 1 or a 0. If it's on, it's going to go out and do the job it's supposed to do. Now, obviously, if it goes out and does the job it's supposed to do, you're going to have the materials there to be able to support the job that you're going to do. And how do you have those materials available? Those materials are available by bacteria breaking down the things that you eat. So here's the whole thing it's an orchestra. Once it says, here's what I need, by turning on the codes, the methylation, it goes out and says, let's get the products to do the job. So it has to have the bacteria there to make the products, and the bacteria has to have the raw materials to break down into the products, the different, all these different molecules that are available, in your whole foods. Your whole foods means it eats from all of this stuff, and all of these things are in your body. They're all available. Now, these ligands grab these little bits and pieces that are being broken down from the bacteria. They just want to grab them whole. And the reason they do, I'm going to go over this in a second. Let me cut off for a second. Okay, my friends, Roger once again, and we are going to be going through some outstanding information. I am going in earnest to start from the basics. We're going to start with atomic and how atoms work and subatomic molecules, and that's going to lead us into electronics, and then how all chemistry works. And talking about that, I started to look into hemoglobin because it has oxygen involved attached to the iron molecules. All right, so it's a little confusing at first, but don't worry, I'll make it very, very understandable. It's not hard to understand. In your blood cells, you have a lot of iron. All right, you ever hear of somebody being anemic? That means they don't have enough iron. Well, they don't have enough oxygen, because those are the things that bind oxygen. Now, let's go through this. This is by a guy, this is in LinkedIn. This is search new jobs for, well, I don't know if this guy has anything to do with it, but it's from Chemistry Explained, Foundations and Applications. All right, now, what's the story here? You've all, I'm sure, heard of that there's hemoglobin in your blood. And what is that? Hemoglobin is iron-containing protein found in the blood in nearly all vertebrates and many invertebrates. And because the oxygen is a combustible item. It transports oxygen from the lungs and gills of animals to the tissues. 4 polypeptide chains. Forget all that stuff. The rest of it is alphabet soup. Doesn't matter to you. What you need to understand is the hemoglobin in your body has iron. You need the iron. Now the iron binds to oxygen. Well, what does the oxygen do? So what about the oxygen? Well, the oxygen keeps you alive. You don't have it, you're dead, first of all. But what it does is each ferrous, which they're all ferrous, iron, I don't know what they say, each iron within hemoglobin provide, well, there is different types of, there's Fe2, which is ferrous oxide, ferrous, we'll worry about that later. There's FeO2, O2 and FeO2O3, I believe that's what it is. And each one of these ions provides one binding site for an oxygen, which is O2. That's the way oxygen comes. It comes as a diatomic molecule, now, in its standard form. Now, thus, a single hemoglobin molecule has the capacity to combine with 4 molecules of oxygen. So just a single hemoglobin can grab onto 4 molecules of oxygen. Well, the oxygen are binding sites to bind to other molecules. It's not just the oxygen. Here's the key. All kinds of other things try to compete for those sites. And when they say they compete, I say they're assigned places according to what you need if you have the correct bacteria creating the correct enzymes to create the correct products to go to these binding sites. That's my statement. Okay, I got to be honest with you. I don't know where I left off or what I actually covered so far, but if I go over something, well, I'm sorry, but I hope I won't. Now, I'm going to be making some colloidal copper. And metallic copper, it's an antimicrobial surface. Very, very strong against everything. Bacteria, yeast, viruses, rapidly killed by copper surfaces. Contact killing. And it goes through a very interesting, and obviously the times are correct. Now, this also has a lot of bioactivity. Look where it is. All right, we got copper, Cu, copper, silver, gold, oxygen, sulfur. This is the kind of things that are reactive in their orders. Now, let's go a little deeper. All right, so I'm looking into Mercury, and because Mercury, I found out something very interesting. I've been working with autism people and people that have all kinds of disorders, and your body has DNA in it. DNA is literally programs. It's literally programs. And there's something called methylation. When you have methylation, it goes into a certain chunk of that DNA, clicks it on, and then that DNA activates where they call the junk DNA, which is 98% of your DNA that they think is junk. That's these islands of program codes that are out there, just sitting and waiting. They don't do anything until you activate them. And then once you go into the strand of DNA and say, okay, this chunk over here, turn it on, the C and the G turns to a 1 instead of a 0. Next thing you know, this gigantic island of DNA out in the never land of stupid DNA turns on, and it goes to your immune system and says, here's how we're going to deal with this issue. That's what's going on. Now, neurotoxins are methylmercury. Say methylmercury has a neurotoxic effect. So I'm thinking, well, how does the mercury affect it? Because methyl and mercury, methylation and mercury. So I go looking at it as, because methylation is literally the programming for the brain. That's the thing that turns it on and off. So I start searching on this, and that brought me to the colloidal silver. And this guy sounds very authoritative. He's talking about, and this is kind of new to me, so I can't say one way or the other if he's 100%. But he's talking about the copper and the magnesium. They all work together. Copper, zinc, magnesium, they're transition metals. What does a transition metal do? I've talked about this a zillion times, and I probably already talked about it in this video, but I'm going to talk about it again. What it is, a transition metal is a molecule, is an atom. All right, it's one of these elements that's in this light blue area, in this darker blue area here. See, transition metals. And they have open little spots on them so they can hook to things. Literally, that's all it is. They can transition from this molecule to that molecule very, 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 very easy. Give me an electron, no problem, come on over here. Boop, it goes. He says, well, I'm going to throw you out of here. That guy's got more electrons, and boop, it goes back. They're reversible. All right, that's how your body works. It gives and takes, obviously. Now, there is certain requirements for metals to work with other metals, because that's how it is. These are not just things that are made and they never change. They are always continuously changing in your body. They're changing. It's called reductase, and the molecules crack and add to something else. That's how your body grows and repairs itself and gets energy and all of that stuff. So I would look at this. It's how to make your own colloidal copper. I'm going to make some. I'm going to show you how to make it. Very, very simple. Just two positive and a negative to two pieces of copper in, sterile water. You know what I'm talking about. Evaporated water. I can't think of the word. Anyway, because you can't have any extra minerals in there. But then he throws in a little, I think he said ascorbic acid. No, not ascorbic acid, citric acid, to give it some little polarity, because the type of water has no ability to make electricity flow. Because you need these little impurities that have polar molecules. I know water's polar, but it doesn't work the same way as these other molecules where they can flow. Anyway, it pushes all these little blue particles into the water, and then you take that and you can put it on your skin. It's a good video. I liked it. So anyway, that is what I was looking into. So I got into the mercury, because mercury is nothing more than blood with sulfur in some kind of a liquid, and then a bacteria. The bacteria creates the reductase that creates the mercury. I'll show you. Okay, because they can make gold too. Here, I'll show you the gold first, and then we'll show you the mercury. All right, you see this? This is bacteria. These bacteria digest toxic metals, poop out tiny gold nuggets. Well, what are they doing? They're transmutating. They're transmuting these elements, these different elements, and they're literally nibbling away at the nucleuses of these and dropping them down to some other molecule or atom, really. And they do. It's called reductase, and it says no other life form on our planet has infiltrated every environment successfully as a minuscule single cells of bacteria. They're everywhere. They do everything. I mean, I'm telling you, if you don't have bacteria in your body, if you're going to fall over and die instantaneously. Amongst their many roles in life on Earth, it turns out some of these microbes are also experts at purifying precious metals. They purify everything. They use them in the waste treatment plants to get rid of everything. So, but anyway, they poop out gold. Now, what else can we learn from this? Hold on. Okay, so I decide, well, where does this toxic neurotoxin come from? Well, cinnabar, it's a toxic mercury sulfide, they say. So here we got the mercury, and then we got sulfide. All right, so, and then it goes on further to say how it's formed in well, here it's hot water or so forth. Here's what it says. All right. Cinnabar toxic mercury sulfide with a chemical composition HGS. It is the only important ore of mercury. It has a bright red color that has caused people to use it as a pigment and carve it into jewelry ornaments, drinking cups. It was used in actually medicine until not too long ago. Now, geological occurrence of cinnabar. Cinnabar is a hydrothermal mineral. Okay, so what does that mean? Hydrothermal, water, hydro, thermal, temperature. Okay, so hot water areas, cinnabar is a hydrothermal mineral. So this cinnabar, this red stuff, is in hydrothermal areas that precipitates from ascending hot waters and vapors as they move through fractured rocks. Now, but before up here they said mercury sulfide. All right, so we know we got sulfur in here. So that's the key in these temperatures above about 200 degrees Celsius. Usually forms in rocks surrounding geological recent volcanic activity, but can also form near hot springs and fumaroles. Now, what causes hot springs, and what causes fumaroles, and what causes volcanoes? In my world, they are decomposition of something. It's like compost. It's like a colon or something like that. The guy's colon of a giant, it's such a giant pile of poop that's going to heat up and overheat. They're covered up. They're locked up down there. The bacteria reductase goes in there. It creates a lot of heat. All right, so here's the cinnabar, and this is where the mercury comes from. Now, if you looked at this, you understood what you're looking at, you're going to see the biology in there. Now, this, I think, could be cinnabar. I don't know. It could just be a little ball of blood. But it's pretty darn close. Now, this is all biology, and it's being changed by several different things. There is thermal sulfur bacteria. It loves hot thermal springs, and it's sulfurous. Now, we know that mercury and sulfide is what we're ending up with. This is mercury. So we got to look into the bacterial reduction of all different molecules, because that's what the alchemists did. They didn't use some special woo-woo-woo. They used bacteria. It's come to me now. That's the whole thing with alchemy is bacteria. You can see, you can make gold, you can make anything. You just have to have the right bacteria working with another set of bacteria, and you start with some kind of base material. You have to have a base material to start with, obviously. And once you have that base material, then you can manipulate it. Now, I would think that the higher up the base material was, the easier it would be to reduce it, which is where you have mercury way up here. Well, you got mercury here, and it backs it down to gold. When I say way up there, it's right next to gold. But you could take mercury. And Isaac Newton said, give me mercury, I'll give you gold. They know how to do this. And I'm sure that this certain bacteria, if you worked it through mercury, you'd end up with gold. Now, let me show you what cinnabar looks like in some of its more exotic forms. And again, cinnabar is where you get this. Well, here's right here. I don't have to go anywhere special. It's right there. That's blood. That's blood. And what happened there is the same thing. Hold on, I got something around here. There it is right here. Let me put a little water on it. All right. What happens... Oops. I got to clean this place up. All right, now, what we got here is, that is biology, and that was a blood vessel there, and it blew out the blood, and it crystallized according to the minerals that were in there, and they had affinity to go to the transition metals that are in blood, which is iron, obviously. It's a now. What do we have here? Okay, this is a fingertip. All right, let me see. I'm gonna make sure I don't. Hope that's visible. Now, you see the blood blow at the end there? That blood is let me back this out. All right, that blood is iron oxide. It's red blood. Now this is the bottom of the finger. It's the way it should be right now. It's the bottom. Now this side is the arterial side. And if you could come down in here, and if you could see, and I know you can't, if in the microscope you can, this is where the artery is, and this side is where the vein is. Veins don't blow out, they clamp off. I've shown this a bazillion times with all my other stuff. Now, this side here, usually the blood blows out right here, but it didn't, it blew out the end. I might have a little bit of flood out here. But anyway, that's, and now, you see down here? Things turn into crystals. All the biology in your body is nothing more than crystals getting ready to grow. All right? In an aqueous solution with the right chemistry and then the right bacteria in there, creating the right enzymes. They don't eat everything. You know, they live inside of what flows out of these things. There's nucleophilic invasion, and there's other kinds of invasions that the blood meal and the bone meal comes out of them, really. It's like the biology part leaves and they stabilize. That's about all the best I can say it. They stabilize. Even like this, that's a toe. That's a toe. He's, oh, Roger, you gotta be crazy. No, I'm not. That is a toe. And that right there is the last bit of bone that's left. I don't know if it's even visible to you. And that right there is the blood at the bottom of the toe, just like this is the blood at the bottom of the finger. They all have the same stuff. This side here, this is the callus on the guy's toe. That's the callus. Just like this one here. Here's one here. Here's another one. This guy, that's this guy's callus. You see it? These look like the toes that are on a Bigfoot. They have these big round toes like that. See? But that's the guy's callus. And that was where that fingernail or toenail was. Now, and I have so much of this stuff. It's DNA certified and bones and and everything is extremely exquisitely articulated. The cartilages are there and all of the wrapping of the bones and every single thing. And the bone goes away. Like this one here. See that? That's the only bones left in this one. I hope you can see these things. I never know if you can. But that's the only bone that's left in this one. It just goes away. Let me see if I can show you that other one. Oops, where is it? This is the bottom of the toe. That's where the blood is at the tip of the toe. And that's where the bone is right there. That's the last bit of bone in this one. And these are all the metals, because this guy died standing up. Now, all of the specific gravity brings the stuff to the feet. That's why all the gold you find in the feet. Tons of it in certain giants. There's going to be literally tons. Now, this was a small guy, and that's what the metals that was in his body. Now, he might have died, because this is pretty heavy. This is a lot of metal in this. Now, he probably died standing like this. This was the low one. And this one, because usually they have 1 foot out, and 1 foot's missing, just gone. This would be the one that would be gone. It's deep, trying to push himself out. This is what I have found, I think. 1 foot is pretty much not above, but not really below the surface. Then you get one is gone. but I know where it is. They're underneath, and you can usually see something that would indicate like a little red spot or something above where there might have been, some kind of biological, conduit going down. Anyway, they are what they are, and Devil's Tower is a foot. Case closed. I'm telling you, it's a foot. No question whatsoever. So it's really time to investigate this stuff and look. So now I'm going to go a little further, because wait till you see what I found out about this. I decided, well, where's this mercury coming from? Where in the body? Because I know that all of these things are body parts, and they concentrate different amounts of things in different areas to do different types of chemistry, because the body requires it. Acids, salts, different, everything in between. Now, so I find out that is in, I think it's Spain. Hold on, let's go to that. Okay, my friends, I just got another email from this guy who claims to be a hotshot geologist, and so for this I will use a turkey feather. Usually I use a hawk feather. Anyway, this is a giant fish, and this is a giant dragon. I've shown it a bazillion times. And if you come in on it, you can see that is a giant fish, whoops, and above it is a giant dragon, right there. And he spit out a bunch of stuff that is right there, which is extremely nasty stuff. As nasty can possibly get. And it destroyed the fish's back, and his fin, as you can see, ate into his vital flesh in that area after combusting here. Now, he was slayed, however you want to call it, with what looks to be a very sharp, whatever it was, that cut his neck. Now, and that is his neck. And it's dragon scaled all the way up his dragon scaled throat, and it is called, I think it's called the Draco Dragon River area. And this is the fluty stuff that is on the side of a dragon, and that dragon runs all the way almost to the Mediterranean. It is North Africa. Now, this I have shown to this guy, Paul, and he is absolutely insane and very, very upset about this. And all the other things that I've shown, and assaults me regularly. So we're going to discuss what he has to say, what I have to say. He's a professional, and this is what you're going to learn, kids. From the professionals, this is how they react to reality. If you see what you see, and all the other things I have, DNA tests, of course, he can't accept that, and I mean, it's just a million things. So let's go forward, but it's a new world, it's a new way of thinking, and the only way you're going to get to think it is to follow me here, because it's not allowed. And this is, and there's so much that you're going to be able to do with your life, with the knowledge I'm giving you. I'm serious. Your life is going to be so limited, and so, I mean, it's just going to be a life lost to continue on listening to the people that have been teaching you, like this guy, Paul. All right, so let's continue. And I found this 100% all the universe. I'm sorry, my friends, and I'm not going to be mean about it anymore. It's just a fact, and it's time that we have to change it. And it's up to you to share this stuff and raise your voices and say, I don't want to live in this world that's just fantasy world, just to support the professors and so forth. This is not the way it's meant to be. Okay, I made a bunch of statements, as I do on all my videos, and I show the evidence, and he's showing, oh, I showed you all these things, and you have no idea what you're talking about. I have provided you with undeniable evidence in the past. Your claims of dragons and fish in Morocco are nonsense. Well, if that's nonsense to him, I don't know what to say. And he this is over and over and over. This is never ending. I have blocked him, and I still have him blocked, because he's just anyway. I even sent you a cross-section clearly providing that they are the same rocks on either side of a cyclone and all these. It's just nonsense and nonsense. You have shown you have no idea about what you talk about. You pointed pictures and best chalk outcrops. No, they're kaolin clays. And claim it is kaolin. Yes, I claim that because that's what it is. It's skin tissues. There are areas, yes, that are from sea creatures, absolutely no question. And there are areas that are kaolin clays, absolutely no question. And the kaolin clays are skin clays, absolutely no question. And he's, he goes on and he's saying I'm taking advantage of children. Listen to this. I've seen you point at a line on a rock and claim it was sill, proving you have no idea what a sill is. Pointed at a picture, Giant's Causeway, which is from Giants, absolutely, and claimed it was carbonate basalt. I never claimed. I don't know what he's talking about. It is, anyway. That is like saying something is a solid fluid, clueless. You make videos throwing in words that you clearly have no idea what they mean, and then you suggest that children currently out of school should disregard what their teachers say. I absolutely say that, yes. No question whatsoever. They should disregard what their teachers say. Yes, I say that. And believe you, and they should believe me because I have shown them the evidence that supports people like you are not honest, or you're either a complete fraud or you're so incompetent that I can't believe you can continue to breathe oxygen and be that incompetent. I really can't. I don't mean to be insulting, but I mean, for goodness sakes. All right, so it says is that you're taking advantage of young, impressionable minds in a totally irresponsible way for your own ego. I think somebody else's ego is what's bruised. Because, anyway, so yes, let's discuss over email what you are wrong, and I'll demonstrate it clearly. Well, all he's showing me pictures of things that are meaningless because they're wrong. I'm not going to do a Skype or a Zoom. First, based on my previous experience, I have no expectation you have any plausible evidence. I have all the evidence. He has nothing for your case, and will just resort to insults. And secondly, I am a professional geoscientist. I have to ensure that I would never put my company or the companies I work with, most of whom are household names, in a potentially embarrassing position discussing 1,600 kilometer long dragons. Well, yeah, Paul, you're going to be in an embarrassing position. I can tell you that, my friend, with someone who has no clue about geology or that particular area and can be disproven in 10 seconds would be pretty high up the list of things that would be considered embarrassing. Well, where do you see the height of embarrassment that you will hit, my friend? That was nice to him. See, hi, Paul. Nice to hear from you. You're right. It just deserves discussion. Anyway, that's the deal. And kids, I'm telling you, I love you. And if you want to listen to your teachers, that's fine. But I would ask them to look into what I am presenting. Ask them if does it have any merit. Do you see anything here? And can you explain why it has no merit? That's what I'd say. Because right away they're going to say like him, oh, no, you have to look at this book. And that book is obviously a book that's wrong. Because everything's wrong. I'm telling you right now, if you show me something that's correct, I would like to see it. Now, I'm going to show you, well, there's things here and there that obviously are correct. But I'm saying that the underlying principles of everything, because the atom is not correct. They don't understand rocks. It's obviously now that they don't understand the medical needs of the body, even in the medical industry. Once you don't understand the atom, they don't understand light. They don't think light comes from the sun or something. I don't know how they think light gets to the earth if it's not there. That's the dark energy and dark matter that's coming to us. They're particles. I've shown all this. They can't understand gravity because so much is so wrong that you can't even understand how something comes to the ground. But they'll go on and talk about wormholes and things like this guy. Anyway, let's just go forward. Don't take a bad attitude towards this. Don't take a bad attitude towards these people. This guy is you know, I got to be honest with you. I'm not really. He's not going to get any Christmas cards from me. But just don't be nasty. Believe me, I have just apologized to everybody in the whole world. I just got so nasty one day that I just let loose, and don't ever do that. I had to apologize, and I do apologize. And this guy he sent back, he says, "Holy smokes, nobody ever apologizes on the internet." Well, you know, you guys be nasty, you throw out insults, and you just don't mean to be that way. But, you know, I've been, if I had to pile a pile of insults I have taken, you know, I could go way past the Empire State Building. Anyway, here goes. All right, so you know we're looking into Mercury. So I decide I'm going to look into where the largest Mercury deposits are. 25% of the world found in Almaden, Spain. So here I am swishing around down there. And sure enough, what do I see is this. I say, well, what is this all about? What is these spots here? What is this little bumpy things going on? So I decide I'll take a look at it because I think I understand what's going on here. Now, I look at these scars and I think, what the heck is this all about? Are they plowing that up for new planting or what's going on? Which they might be. Or are they just something wrong with these spots? They're not growing correctly. Now, I have a feeling that it may be that these particular spots right here, this particular area, is the same as this particular area right here. This is a tendon. And there it's a gluey area where it has these mineralizations which may not grow anything. I don't know. Maybe that's what's there. I have no clue. I'm just looking. Now, that's where the tendon runs. Now, there would be, it depends on if the tendon's stretched or whether it's pinched, what kind of effect you would have. But I started looking to see, because they, like again, you have an abrupt transition anchor right here. And you have some gluey stuff down below, and I think I see that too. You see this gluey looking stuff? That's what I see in the muscles. I mean, I can show you one I have right over here. It's just virtually the same. It has this, and then it broadens out into a muscular thing, and originally you have this kind of mineralization until it gets into the muscle fibers, and I believe that's what we're seeing, something like that there, tendon, muscle fiber, connective tissue. And nobody cut these in here, these different white spots like this. Absolutely not. And I could see, you see these reddish areas, that's where there's biology. I could show you the biology spots. There's a biology spot right there. This could be looked at. This can be tested. See right over here, see yellow and red? That means that there's probably vein and artery blood there. And again, nobody has cut these cuts into these ridges here. And they're one right after the other. Boom, boom, boom. And these are the lump, the lump, the lump, the lump. Now, is that a tendon? I don't know. Is it a muscle? I don't know. But I'm going to tell you one thing right now. It's biological. And if somebody went out there, they would find this. I mean, look at it. That is not somebody did that. Just did this and then they did that and they did another one over here. Absolutely not. I understand this stuff very, very well. And this is broadening out into some kind of maybe a bigger muscle or whatever. And it's everywhere here. This is not just, and it's all over the place. Now, this is where they have the most, right over here, Almaden. Now, I looked over here, and of course it has the same sort of things here. You know, over here they're doing, I don't know what they're doing here, but that's, nobody made these things here. Look at this. So what I need to see is what is the chemistry involved in this? Are these like scales and so forth? Because there's obviously something. What they are at this point, I haven't been able to determine, but there's, well, if you can't see, there's a pattern to these things. And what happens with the things that I'm talking about that I can't say the word, which is this, they concentrate biology. All right? Biology concentrates minerals and metals in different areas. It's just the way it works. There's different chemistry in different parts of the body. Now, I don't know what dragons had and giant fishes, but I can tell you what, that's a giant dragon right there. All right, if anybody wants to contest any of this, come talk to me, because I can contest to anybody all day long. That's a dragon. Just go up here. You can't miss it. It's not hiding. It's not something you have to dig around for. And there's a story about this that I'm not going to bother telling again. I told it a million times, but anyway, and it's attacking the fish. And they knew about this in the ancient past. I know they knew about it because I can show you this. Somebody sent me this. I don't know. It's what it is. I don't see it. This is a griffin, really. So, oh, that's a griffin. Well, it's close enough. It's not an accident. Now, this is a word you're going to hear in a moment, leukocyte. And what does it do? It's a white blood cell. And what does a white blood cell do? Well, it's a colorless cell. It circulates through the body just like blood and in the body fluids, and it's involved in counteracting foreign substances and diseases. It's a white blood cell. Now, there are several types, all amoeboid cells with a nucleus, including lymphocytes, granulocytes, monocytes, and macrophages. Now, they are the repair guys in your body. Let's take it another step. All right, so now you know what a leukocyte is. Now, what is hydrogen sulfide in redox biology? Well, this is 2015, and we know what hydrogen sulfide is because it comes from hot springs and in these electrophiles. And apparently people soak in it, and the mechanism of anti-inflammatory effects of hydrogen sulfide. The ability of hydrogen sulfide to reduce pain and inflammation has been long recognized for centuries, though not until recently understood on the molecular level. Natural sulfur springs have long been used as a means to alleviate the symptoms of an array of inflammatory diseases. Inflammatory diseases means something is irritating you, or inflaming you. Why? Why are you getting inflamed? And what is getting inflamed? For me, it's my back and so forth. Sometimes I get sore. Now, I've just run across some very important, well, I hope it's important. It sounded pretty good to me. Information, I think it's in this video, about the copper, colloidal copper, which I understand that there's different metals in your body that are critical, and they're right here. You got copper, silver, gold, zinc, nickel. All of those things are extremely critical for your body to do business. And apparently the copper is not in your body as, I don't know, this is all new to me, that particular thing, but I'm going to look into that copper. It sounds pretty interesting. But this, they're talking about, they're finally understanding the molecular level issues. So it says, in the past decade, considerable progress has been made in identifying a molecular mechanism underlying the anti-inflammatory properties of these molecules, all right? Hydrogen sulfide. Now, one of the earliest events is an inflammatory reaction is the recruitment of leukocytes, which is what we talked about, all right, to the site of the injury. They go there to fix things. Those are the white blood cells. And there's a bunch of different ones, and they react differently to different problems. Now, this involves upregulation of adhesion molecules. Now, what is that? On the vascular endothelium and on the leukocytes, adhesion of the leukocytes to the endothelium. And then migration of the leukocytes into the interstitial space. Now, we've shown the interstitium, which is, there's a bunch of little balls and straps and so forth, and it's areolar tissue and so forth. There's a bunch of different terms they have for it. But it's a barrier inside between your meat, between the outside layer of your body and the inside layer of your body. That's how I'm going to say it. That could be right, could be wrong, but that's how I look at it. Now, so there's an upregulation of adhesion molecules. What was an adhesion molecule? Those are something that makes something stick. All right, the adhesion of the leukocytes. These are the things that are the white blood cells to that endothelium, and then migration of the leukocytes into the interstitial space. So they adhere to the layer of tissue that they call the endothelium, which is in between those layers of your body, and then they migrate into the areolar tissue, from where they move up a chemotaxin gradient. Now this is just unbelievable. So now what do we have? Leukocytes. These are the white blood cells. They come up and they go into the chemotaxin gradient to the site of injury or infection. And I believe they are moving along the nerve corridor now. So they're moving somewhere, I believe, through the fascia system. And there's a lot to be looked at there. Now, hydrogen sulfide plays a key physiological role in regulating these processes. Indeed, H2S is a tonic inhibitor of leukocyte adherence to the vascular endothelium. Indeed, H2S is a tonic inhibitor of the leukocyte adherence to the vascular endothelium, as is evident from the observation and administration of inhibitors of this stuff results in a very rapid increase in leukocyte endothelium adhesion. So this stuff stops them from sticking. H2S. Now, this process is mediated, upregulating of intercellular adhesion molecule and P selection on the endothelium. It sounds like to me that the H2S is a tonic inhibitor. It inhibits the leukocyte adherence. Now, I would think the leukocyte adherence would be good. Let me look into this. Well, there's your answer. Leukocyte adhesion deficiency. So you got a deficient with your leukocytes. They're not sticking. Is a primary immunodeficiency disease characterized by the inability of leukocytes to migrate from circulation into sites of inflammation, resulting in recurrent bacterial infections. So you can't get rid of the infection because your leukocytes are not stuck to. I call that membrane-bound bacteria. Now they call them leukocytes, white blood cells, and they might be. I might be totally wrong. They call them white blood cells, and I guess they are. But what I believe is maybe the white blood cells have to stick there so that the bacteria can grow there. That's what I'd go. The white blood cells are the signals possibly for the bacteria. Because I'm telling you, I think it's all bacteria. Now, because the bacteria, unless these leukocytes can create all of these different enzymes, and I don't think so, because they only talk about 4 different types of leukocytes that I saw. Now, there might be a whole variety of them, but I can tell you one thing, there is so many bacteria that if a leukocyte goes out there and says, hey, I'm going to need some lactose out here, give me 6 of these and 10 of those and 172 of that, and they come out and they go to that leukocyte location as a signal. That's the way I'm going with this. And it goes on and on. There's a lot to understand about this. It really is. But see, they're bacteria. Let's see what it's got to say about bacteria. Migration to sites of inflammation of infection and phagocytosis of the bacteria. The binding of neutrophils to these activated cells is also mediated in part by selectins, not affected in leukocyte. I don't know. I'm telling you, I'm going to go, well, here it is right here. It always gets into here somehow. Gastrointestinal manifestations of primary immunodeficiency, which is this thing. Here we are again, leukocyte adhesion deficiency, characterized by impaired phagocytic function secondary to deficiency necessary for cell migration. Let me look into this. Apparently, your gut is the cause of all these things. I'm telling you. What's this about? Antibiotic therapy is required for recurrent infections. Prophylactic antibiotics are sometimes necessary. That's the prebiotics, probiotics. Meticulous dental care, oral hygiene are recommended so you don't get infected through there. But you should have some bacteria in you that fights all these things. And I look, here's another. Gastrointestinal complications include intraoral infections, periodontitis, esophagitis, gastro this, and appendicitis. All right, that's because that's where all this gut bacteria is stored, necrotizing and abscesses. Get your gut in order. That's my recommendation. Okay, I think by now I've shown you that I believe mercury is from blood and it's from sulfur in this certain temperature. It's a pretty high temperature with blood with sulfur. Now, and I'm saying that has to have some bacteria there that does the reductase in this extreme environment to end up with mercury. And guess what? I go looking into sulfur springs, hot springs, and this blows my mind. I'm telling you, every time I look at something, it just goes deeper and deeper into the knowledge realm of understanding how these things work. Acidic environments and acidophiles. Now, this is where you get sulfur. The acidophiles are the bacteria that live in this extreme environments of heat and acids, like lactic acids and sulfur and acidification. All kinds of things are going on here. And they're bubbling and hot and all that. Acidophilus microorganisms can be found in all three domains of life. This is the one I've been studying. This looks different. Acidify. Hold on a second. Let me just get what I got here. Hold on. That's not the same stuff. Okay. Yeah, I'm doing this real time. I don't do any scripts, so when I run into a thing like this, we're going to just investigate it. I've been looking into acidophilus. This is a probiotic, and it absolutely, the well, here it is. Lactobacillus acidophilus. Okay, that's how it's. But there's all kinds of lactobacillus, but this is the key one. Now, this is talking about acidophilic, which is acidophilus microorganisms can be found in three domains of life. Okay, so that's the key. This one here, the acidothiobacillus, thioxidias, forget all this alphabet stuff. Bacteria cannot grow at pH values above 4 to 6. Okay, so our body has to be a 7.35, 7.45. If it's outside of that range, you have acidophilus, or you have the other thing, acidophilus, or you got too much salt. Now, so anyway, that's the difference between, but it's the same sort of stuff. But this is an extremophile version of this. It's all coming to look, optimal growth temperature, 60 degrees Celsius, above pH 4, because there's going to be certain characteristics in that environment that are going to aid the production of a certain breakdown, which is going to be mercury in this particular heat and acid content and sulfur and all of that, if there's a lot of blood, because the blood has all of this stuff in it. If you can get it converted, you get whatever you want out of the blood, really. There's only, and I'm going to show this in my experiments with making the colloidal stuff. if you grow plants, all you have is blood meal and bone meal. You feed those two things to your plants and they grow. Because that's all that's in the things that we throw in the, bodies end up going in the ground. I'm telling you, I was thinking about, these burials and so forth, and I suppose burning somebody out, combusting them, they go back into the environment. But anyway, that's a whole other issue. I always get off on these crazy things. But anyway, I'm talking about this particular set of circumstances with a lot of blood can create mercury. Now let's go see where that exists. 25% of the world's mercury comes from one spot. I'm telling you, I am getting old, so this could be that I am losing it. I'm serious, but I also have a lot of things rolling around in my mind. Now, before I said you got acidophilus or you got something else, which I couldn't remember, which was acidosis or alkalosis. That's high acid or high salt. So anyway, I'm just explaining what I said before was wrong. Now, but this is the key. In acidosis, the pH of the blood is abnormally low. So the pH is low. You're more acidic in your blood. Now, and again, 7.35, 7.45. So it shouldn't be lower than 7.35 or you go into acidosis. Now, it's associated with diabetic ketoacidosis, lung disease, severe kidney disease. Now, I'm going to start looking into this. Apparently, some people have continuous acidosis, and they get this. Now, the opposite acidosis is alkalosis. That's alkaline. You're in the range of saltiness, in which there's too high of a pH due to excess base, which is salts, or insufficient acid in the body. So, it gives me another spot to look, but because I really haven't looked too deep into and I hear people saying all the time, you got to take bicarbonate of soda, and you got to take this, and you got to vinegar, and all these things, and they're different acids and bases. So this is pretty interesting. Anyway, I just wanted to make the point that it is. Anyway, you know what I'm talking about. All right, I'll leave you with this. They're just laying on the surface. I didn't pick that out. Well, somebody probably dug them up at some time, maybe. I don't know. But they are on the surface now. Let me see. See this one? You see, that's a fingertip. I'm telling you, that's a fingertip. I probably smashed it up. But I think you can see some biology in there. I mean, I know these for a fact. I've looked at the ones I say are anyway, let me show you another quick shot, and then we'll call it quits. All right, this is just one of those. I mean, they're everywhere.
See, this is New England.
They just run down, and I got them all the way across my fronts in the front.
Rock fences to the right.
You got fields going this way, fields going that way, fields in the back. They were all
of these things. Every one of them is something. I mean, it's the rocks are not just nothing. They can all be examined, and I can examine a hell of a lot of them.